So chapter 48 isn't going to focus on a specific system. We're going to try and talk about neurons and synapses as well as signaling to get us prepared for chapter 49, which is about the nervous system. So that picture you saw on the first slide was showing you a cone snail. They are able to kill their prey using venom that disables neurons. Neurons are your nerve cells that are able to move information throughout your body. Two types of signals that neurons use for communication: electrical signals, which are long distance, and then chemical signals, which are short distance. The nervous system has to interpret those signals, and it is able to do that using some complex paths and connections. This processing will take place either in simple clusters of neurons, which are ganglia, or more complex organization of neurons, a brain. Okay, this was studied a lot um, in this particular organism because you can see it has the squid, because it has really, really large axons. So that made it a lot easier to be able to examine what happened. Okay. Neuron organization structure is going to show us how is going to play a role in reflecting function in that information transfer. In other words, neurons have different shapes depending on the type of neuron that they are representing. We have three overlapping stages in which nervous systems are able to process that information from their neurons. We have sensory neurons, which are the ones that are able to detect external stimuli, internal conditions. They move information along to the integration stage, where interneurons are able to integrate sensory input and motor output. These are typically in the brain or again in your ganglia. And then in the motor output, we use a third type of neuron, a motor neuron, which moves signals out of these processing centers. And the motor neurons, when they transmit these signals, are able to trigger muscular and or gland activity. So there's a picture at the top showing you the sensory neuron and how it overlaps with the interneuron, which overlaps with the motor neuron. And then the pictures at the bottom are showing you the shapes of those different neurons. You can see how the sensory neuron, its cell body, which contains its organelles, its nucleus, is in the middle, while the interneuron, it's actually a part of the axons. And then with the motor neuron, it is right at the top along with its dendrites, but that the majority of the axon is straight and able to connect with whatever its target is using the hillock. And then the red that you see in the picture above, we'll talk about that in a little bit. Those are myelin sheaths, and they help to improve conduction rates. So, nervous system, again, we're going to spend a lot more time on this in the next chapter. Animals have a pretty complex nervous system that consists of two parts: central nervous system and peripheral nervous system. The central nervous system is where the sensory input is integrated with the motor output, your brain and your nerve cord. The peripheral nervous system are your nerves that extend into different parts of your body, tend to be bundled together. They're the ones that take the information to the CNS and take it out from the CNS. So your eye is acting as a sensor in this particular case. That information that it is sending is integrated in the central nervous system, and then it is sent out of the central nervous system to the affected, in this case, muscle, through the peripheral nervous system. And then the information is sent in from the eye to the central nervous system via the peripheral nervous system. Neuron structure. Neurons have cell bodies, dendrites, and axons. The cell body, again, has your organelles primarily, along with nucleus and cytoplasm. It's found pretty much in the CNS. There are some that are found in ganglia, and then they have two extensions, two extension-like objects, dendrites and axons. Dendrites are the ones that receive signals from other neurons. The axon, again, is a much longer extension, and it's going to transmit signals. So dendrites receive them, axons transmit them. and its cone-shaped base is known as the axon hillock. Okay. Supporting cells. Vertebrate neurons, most invertebrate neurons require these. They help to provide support for the neurons and help it do its job. They are not involved in conducting impulses, and there are large numbers of them present. They outnumber mammalian brain neurons 10 to 50 fold. We have the glial cells, your astrocytes and oligodendrocytes. We'll talk about those a little bit more later, and your Schwann cells. The glial cells, the astrocytes and oligodendrocytes, the example of it's the one on the left, the Schwann cell is the one on the right, and it's the purple one. Okay, so there you can see all the glias that are surrounding your neurons. Neural circuits, populations of neurons that are connected together by synapses that carry out specific functions after they have been activated. We have large scale brain networks when neural circuits interconnect with other neural circuits. So, individual towns coming together to make up a city. Simple circuits are present when a presynaptic neuron is able to stimulate a single postsynaptic neuron. But there are more complex types of neural circuits, and we'll talk about each of those on the next slide. Sorry, slide coming up after that. So there's your presynaptic cell to the top. The signal is being sent down its axon to the postsynaptic cell where the dendrites are there, and it sends it off to where it needs to go to. We talked about how it could be electrical signals. It can also be at the synapse where the presynaptic cell and the postsynaptic cell interact. It could be a neurotransmitter, a chemical signal that sends it off. More complex circuits, diverging circuits. When one neuron synapses with multiple postsynaptic cells, and we tend to see that with motor neurons. In a converging neural circuit, you're taking in information from lots of different sources and converging it into one output. And that's what we see with the brainstem and respiratory, being able to breathe. Need to connect a lot of information together to make that process take place. In a reverberating neural circuit, neurons send signals one to another in a linear pattern, and then they send it right back, the last one to the first one. Breathing would be an example of a reverberating neural circuit because we don't ever want to stop breathing. In a parallel after discharge circuit, this involves both diverging and converging. A neuron sends input to several neuron chains of varying sizes. So that would be the diverging. And because they're different sizes, they're going to converge on a single output neuron at different time intervals. That would be the converging piece. Continuing firing after the output neuron has stimulated. After stimulation has been ceased is called after discharge, and blinking would be an example because we can blink at different rates of a parallel after discharge circuit. Okay, so those are pictorial representations of those four types of more complex circuits. Sorry, this next section is going to be a lot of chem. Ion pumps, ion channels are able to determine a neuron's resting potential. Between the cells and the interstitial fluid, there are ions that are not distributed equally. They are attracted across the membrane to one another, opposite charges attract, and that is a source of potential energy. And that difference, in which we are going to call voltage, is known as your membrane potential. When a neuron is not sending any signals, it is at its resting potential. Inside the cell, it typically is between -60 and -80 millivolts. And when there are changes in this membrane potential, those are able to act as signals and will cause at times for the neuron to become activated to transmit and process information. So, how do we get this resting potential? When you have a mammalian neuron that is at resting potential, the potassium concentration is greatest inside the cell, and the sodium concentration is the greatest outside of the cell. A while back, we talked about sodium potassium pumps and how they are able to use energy concentration gradients to move sodium out of the cell and potassium into the cell. And those gradients are sources of chemical potential energy. When those ion channels open, they're able to convert chemical potential into electrical potential. There are, because you have a selectively permeable membrane, there are potassium channels and sodium channels. And if you have open potassium channels, you're going to allow potassium to leave the cell, and if the sodium channels aren't open, the sodium and the anions are going to stay in the cell. Anions, remember, are your negatively charged ions. So, because you have allowed a lot of potassium ions to move out and you've kept the sodium and the anions in the cell, you have a negative charge that builds up, and that becomes the predominant source of membrane potential. Okay, so there's your different concentrations. We're going to focus mostly on potassium and sodium. A lot of potassium in the cell, a lot of sodium outside the cell. Again, if you move potassium out, you're losing positive charge, and that's what causes that negative potential to develop at resting potential. Okay, so there's your sodium potassium pump. You can see how you have sodium ions moving out and you have potassium moving in. And then when that pump is open, it allows potassium to move out and it can allow some sodium to move in. But we see a lot more potassium moving out than we see sodium moving in. So basically, look at this as two chambers. The potassium chloride concentration, the source of potassium ions, is greater in the inner chamber and less in the outer chamber. So it's going to move down its gradient to that outer chamber, going from higher concentration to lower concentration until both electrical and chemical forces are at equilibrium, that they're balanced. And because the chloride stays in the inner chamber, you've got that charge attraction for the two sides. And when you reach equilibrium, both the electrical and the chemical gradients have to be balanced. We can actually measure or calculate this equilibrium potential for specific ions using what's known as the Nernst equation, which is giving you that the um, potential or the voltage of that particular ion is equal to 62 millivolts times the log of its outside concentration divided by the inside concentration. And so you take the log of that ratio. For a potassium ion, that equilibrium potential will be negative since you have more ions that have moved out, while sodium, having not left, will have a positive potential. And when you are in a resting neuron, the currents of those two ions are equal and opposite one another. So that resting potential, because they're changing at the same rates, is going to remain pretty constant. So we've got here your potassium channel, and you can see how the potassium is moving out. And while sodium is not being able to move out, there are some channels that are not voltage gated, so some sodium will move in. We see that the membrane is going to have a positive cell potential for sodium. Action potentials are able to be generated by the signals that are conducted via these axons, and they are able to take place because of these gated ion channels that will open and close in response to stimuli. When the potassium channels that are gated open up, potassium leaves, and that makes the inside of your cell have a more negative potential, and we refer to that as hyperpolarization. When the magnitude, the size of that membrane potential increases, it's becoming more negative. You can have some ion channel changes that will cause the membrane to be depolarized, which would reduce the magnitude of that membrane potential, making it become less negative. So, if you open up your sodium channels and you increase the sodium concentration inside the cell, you're adding positive ions, which are going to help to balance out that negative charge from the chloride, and that would reduce your overall cell potential. So, sodium potassium changes can both influence hyperpolarization and depolarization. Graded potentials are polarization changes where the magnitude of your change will vary depending on your stimuli. Um, Depending on how far they are away from your stimulus source, you're going to lose that magnitude as it gets further away. It's going to decay. So these aren't the actual nerve signals, but they have an effect on generating them. Regardless of the stimuli that has played a role, if you are able to generate enough of a depolarization, if you are able to reduce that negative cell potential to what we call its threshold, about negative 55 millivolts in a million neurons, that neuron is going to go through transmission. And once that transmission occurs, it doesn't matter how strong that stimuli is, even if it depolarized it more than that, as long as it depolarized it up to that point, you're going to have an action potential generated. These action potentials are not one-off events. They can occur in adjacent regions along that membrane. They can't occur halfway. They either happen or they don't. And they are able to send signals. Remember, we talked about how electrical signals are sent over long distances. Because some of the ion channels in your membrane are voltage gated, not all are. Once that membrane potential gets to a certain level, one depolarization will lead to additional channels opening, which can lead to additional depolarization. So we see that positive feedback mechanism in place. So, how an action potential works. When you're at a resting potential, the reason the cell potential is not the same as potassium's cell potential is because there are some channels where sodium is able to passively move through into the membrane, and so that causes your resting cell potential to be just under potassium's. Um, once so the voltage-gated sodium and potassium channels are closed when you're at resting potential. When you have some sort of stimuli come along that opens up the voltage-gated sodium channels and you increase your concentration of sodium ions inside of the cell in the cytoplasm, inside the cell membrane, if that depolarization meets that threshold value, you have an action potential occur. There will be both a rising phase and a falling phase. With the rising phase, once the threshold is crossed, the depolarization will cause the most of the remaining voltage-gated sodium channels to open, but it'll keep the potassium voltage-gated channels closed. When that occurs, because you're moving in lots of sodium ions, your membrane potential is going to become more positive. In the falling phase, the sodium channels that are voltage gated are blocked. They are not able to allow sodium ions to come in anymore. But the voltage gated potassium channels open up, and because potassium is going to be moving towards its concentration gradient, it flows out. So that's going to reduce your positive ions, and it's going to cause that potential to become even more negative. The undershoot period is we've got the voltage gated sodium channels closed off, but even though we have gone more negative, sometimes they stay open a little longer than they need to, and that causes your membrane potential to be more negative than what we would expect. Once it reaches a certain point, the potassium channels will be closed and the sodium channels will become unblocked, but they won't allow sodium ions to move out, and that resting potential will be restored. Um, there's a refractory period after an action potential where you cannot initiate a second action potential. The sodium channels are going to be temporarily inactivated for a period of time. So there's pictures of it starting down in the bottom left-hand corner. There's your resting state where sodium's coming in, potassium is not. You have depolarization occur, and you have more sodium. You can see the charges showing up there. You have more sodium coming in, okay? But you can't have potassium going out. In the rising phase, um, we see that we've got lots more sodium coming in, so we're becoming even more positive. And then in the falling phase, the sodium channels are blocked off. Sodium can't come in, but now potassium can leave, which is going to drop your potential. And then the undershoot, where you've gone too far beyond that resting potential, where your sodium channels are open, but they're not blocked anymore, but they can't allow any more sodium to come in, and potassium is still able to leave. And then we eventually reestablish that resting potential, going back to number one. So where these action potentials are conducted is usually at the axon hillock. The electrical current will depolarize a region on the axon membrane. They only travel towards the synaptic terminals from presynaptic to postsynaptic. Because the sodium channels have been inactivated behind them, we talked a little bit about how you can't start that second one off. It can't. This is also done in part to keep that action potential from going in reverse. But we also said how it can move from one part of a membrane to another part, so it can keep generating as it moves across that axon. An action potential speed will increase with its diameter, and vertebrates are able to increase that diameter with that myelin sheath, which is made by glia, the oligodendrocytes in the central nervous system, the Schwann cells in the peripheral nervous system. Um, these potentials get formed at nodes of Ranvier. They're myelin sheath gaps where we can find those voltage gated sodium channels. And the action potentials will move in these myelated axons in a process we call saltatory conduction. Okay. So it allows them to keep moving and it increases that diameter, which speeds up that process. So it's when you have damage to your myelin sheaths and you have more opportunities for the depolarization process to occur, that you can start to run into some nervous system disorders. Okay, so there's your Schwann cells, and you can see there again where the gaps are that you're able to have those depolarization regions along your membrane. Neurons are able to communicate with cells at synapses. Um, the action potentials aren't going to be transferred necessarily between neurons and other cells, but information will be. In electrical synapses, the current is transferred to help to promote additional action potentials from being generated. At chemical synapses, which is what we find for the majority of them, a neurotransmitter will take information across, and that neurotransmitter will help to activate channels. It's taken across in synaptic vessels, and the action potential will help to release it, which diffuses it across the synaptic cleft, and then it's received by the postsynaptic cell. So there you see your action potential coming down the axon, and this particular one is a chemical signal. The synaptic vessel has your neurotransmitter. The cleft is between the presynaptic and the postsynaptic membranes. As that synaptic vessel fuses with the membrane, is released. It's able to bind to those ion channels, and that's going to cause additional depolarization events to occur so that you can have more action potentials. You can also see that calcium is going to play a role as well as that enters the voltage gated channel for the presynaptic cell. So direct synaptic transmission is going to be binding those transmitters we talked about to the ligand-gated ion channels, which will generate postsynaptic potentials. And there's actually two ways you can have postsynaptic potentials. You can have excitatory postsynaptic potentials, EPSPs, ones that will help to get your membrane more towards the threshold. And you can have inhibitory postsynaptic potentials, IPSPs, which will move you further away from threshold, hyperpolarization. Um, so feedback. After release, once the transmitter has been diffused out of the synaptic cleft and taken up by the cells, it can be used to help to generate more of these postsynaptic potentials, or it can be broken down by enzymes. So there are synaptic terminals of presynaptic neurons, and you've got a whole bunch of them that are right next to that postsynaptic neuron. So sometimes you can have multiple postsynaptic potentials play a role at the same time. A lot of neurons have multiple synapses on their dendrites and cell body. One EPSP or one IPSP is typically going to be too small to generate an action potential in a postsynaptic neuron. But if they get it from multiple sources, then those can be summed together and result in additional action potentials. Spatial summation is when you have two or more EPSPs occur at a single synapse so quickly that they are added together. Your postsynaptic neuron doesn't get to go to resting potential. It happens so fast it's not able to return back. In temporal summation, you have it occurring at different synapses on the same postsynaptic neuron. And in that case, they would be added together. Um, so when you add these EPSPs through both spatial and temporal summation, together they may trigger an action potential. And then the same thing would work with IPSPs to counter an EPSP's effect. And so, depending on what all is taking place at that postsynaptic membrane, it will determine whether the axon hillock, the neuron's integrating center, can reach that threshold and have another action potential. So, in letter A, we've got the terminal branch of your presynaptic neuron, and that it is having releasing its electrical or chemical signal on the postsynaptic neuron. It did not generate enough of a potential change, so therefore there was no threshold. With the second particular example, we have that first E1, that first branch of the presynaptic neuron, hitting it twice. Because it hit it twice, it was able together to generate enough of potential to reach threshold, enough of a depolarization event, and the action potential continued. And then the axon hillock was able to move it forward. In letter C, we have two different presynaptic neurons hitting the same postsynaptic membrane. And together, when they were summed together, spatial summation, we have the action potential that was generated from the threshold being met. In letter D, we have E1 um, undergoing its potential and depolarizing the membrane of the postsynaptic membrane. Then we have an I. So now we have that is hyperpolarizing the membrane. And when you combine those two together, you have not reached threshold. So there is no summation event and there is no action potential that occurs. Some synapses, the neurotransmitter binds to receptors that are not part of ion channels, metabotropic. When this takes place, the ion is able to move through the channel via metabolic steps. Um, the neurotransmitter, when it binds to it, will set off a signal transduction pathway involving a second messenger. And so, although it is not going to necessarily generate an action potential, they will take a little bit longer to get going, but they also last longer. So some examples of neurotransmitters. There's five main groups. We have the acetylcholines, the biogenic amines, the amino acids, the neuropeptides, and the gases. And each of these individual neurotransmitters can have lots of different receptors. So there are examples of them, and we'll talk about each of them on the subsequent slides. Acetylcholine is a common neurotransmitter in vertebrates and invertebrates. It plays key roles in muscle stimulation, memory formation, and learning. Vertebrates have both ligand-gated and metabotropic acetylcholine receptors. Amino acids are active both in your CNS and PNS. The ones that work in the CNS are glutamate. This is going to play a role in your long-term memory. GABA, gamma aminobutyric acid, which is present at most of the inhibitory synapses in your brain, and glycine, which acts at inhibitory synapses in the CNS that exist outside of your brain. Biogenic amines, epinephrine, we talked a little bit about some of these earlier. We did endocrines, made from tyrosine, affects your heart. Norepinephrine is also made from tyrosine, and similar to epinephrine, it's going to be affecting your blood vessels primarily. Dopamine is coming from tyrosine. It affects your movements and emotions. Serotonin, which is from tryptophan, and that's able to help relay messages in your brain. And all of these are active both in your CNS and your PNS. Neuropeptides, short chains of amino acids that are able to operate via those metabotropic receptors. They are made by cleaving larger protein precursors. Endorphin, substance P, are two examples of those. They are, neurotransmitters that affect our pain perceptions. And opiates bind to the same receptors as endorphins, which is why they are able to be used as painkillers. Gases. Some neurons and vertebrates are able to release dissolved gases like nitric oxide and carbon monoxide. These gases are not just made on a constant basis, they're synthesized on demand. They act as local regulators in your PNS. They help with smooth muscle cells, and they regulate hypothalamic hormone release in your brain.